It's time for the autoclave. After you've made sure that the bottles are only loose so that the pressure doesn't build up and cause them to explode or the capsule gets stuck on, and you've only filled your bottles part way so that things don't boil over, you put everything in a tray so if something boils over, it'll be stuck in the tray, and you've stuck it on a liquid cycle if you have liquid so that the pressure is going to decrease slowly so that you don't get it to boil because now the pressure's dropped, it's easier to boil. Normally how an autoclave works is what happens is it's really, really high pressure, so it makes it hard to boil, and therefore it's hard to evaporate, and you get to a higher temperature before you get to that gas, so we're able to be kind of like really, really hot steam that's going to kill the bacteria and stuff by denaturing or unfolding all their proteins and stuff. Here's more about how it works and how to use it.
An autoclave is a giant steam sterilizer, and we use it to sterilize things like bacterial media, so the food that we grow bacteria in, as well as pipette tips and glassware, all sorts of different things that we want to clean up. And the way that it does this cleaning up it does this disinfection is by using steam. And not just any steam, but super, super duper hot steam. And it's able to do this because it gets to super, super high pressures. So when we talk about steam, what we're talking about is water in its gas form. In order to go from a liquid to a gas, so to evaporate, the molecules in that liquid have to have enough energy to overcome one another and to overcome the external pressures being applied to them. They have to be able to break free from their attractions to one another and escape as a gas where they can move around freely. Now, under normal pressure conditions, it's going to be easier to evaporate. So you can evaporate at a lower temperature. But if the pressure is higher, you're going to have to get to a higher temperature in order to evaporate, because that high pressure is going to make it so that those molecules are kind of pushed against one another, making it harder for them to escape. And so if you're able to get to a really, really high pressure, you're going to you're going to make it harder for the vaporization to happen. And therefore, you're going to have to get to higher temperatures in order to do this.
And importantly, as we'll get into, we're actually pumping the steam into the chamber. So that steam that we're getting, it's not from our stuff evaporating off. Instead, it's from the steam that we're pumping in. This way, we're able to introduce this really, really hot steam without being at temperatures in the environment inside of the chamber that are actually going to cause our liquids to just like boil off. We're putting this really hot steam in there. The steam is going to meet the surfaces of our containers and things, which are going to be cooler. And then it's going to transfer the heat, as we'll see, and cause the denaturation and all of this stuff. Without our samples just like boiling, we're able to get this really, really hot stuff in there. So it's super cool. I mean, super hot.
But what's happening in autoclave too is you have this closed system. This means that as you have the evaporation, you're going to get to this equilibrium where the evaporation or going from a liquid to a gas is going to be equal to the condensation going from a gas to a liquid. And there's something special that happens when something undergoes one of these phase changes, and that's that the temperature doesn't increase. And so basically, I need to step back and talk a second about heat versus temperature. So these are related but distinct terms. Heat is the actual energy that gets transferred, whereas temperature is the average kinetic energy or energy of motion of a group of molecules. So when you add heat, you're adding energy. And initially, this energy is going to be used by those molecules to break free from one another. So they're going to be using that energy just to break free, not to actually kind of move around more, just to kind of wiggle more to break free from one another. And therefore, because they're using this energy in order to break free, you're not going to see an increase in the temperature. You're not going to see an increase in the average kinetic energy of that movement of the molecules at the bulk level. And so what's going to happen is that you have this thing called the heat of vaporization, which is going to be that heat that's required in order to break free from those attractions to one another. And before you can actually become a gas, like in a process of becoming a gas. Now you can't just gain or lose energy. So what's going to happen is that if you were to go the other direction, which you are doing all the time, especially when you're in one of these closed systems, you're doing condensation, going from a gas to a liquid, as well as that evaporation, going from a liquid to a gas. When you do this condensation, what's going to happen is you're going to release that energy. You're going to release that heat of vaporization. And this is going to allow this big burst of energy to be transferred to the surface that you're condensing on. And so when you take your bottles and things out of the autoclave, you might be a little annoyed. You're like, oh, why is there all this condensation on there? But really, that's just a sign that this autoclave had done its thing, because you can think about when all those molecules in the steam condensed onto that surface, they were releasing huge bursts of energy that were going to then denature the proteins of the microbes and do all sorts of things to inactivate the bacteria and other things that were growing on there. Now, when we're talking about an autoclave, we're not just talking about dry air, hot, dry air. Instead, it's really important that we have steam. The thermal capacity of saturated steam, so basically how much energy can be held in that gas that can then get transferred to the surfaces, this is going to be much higher than that of hot, dry air. So you start by getting rid of the air that's in there, and we'll get into a little more into the mechanics. But basically, there are going to be a series of valves and things where you can pump air, steam in, and push air out, and things like this, because you want to have inside of that chamber, you want to have this saturated environment where it's basically very, 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 very humid. And you have this equilibrium happening, and the steam itself is going to be getting into all of the various nooks and crannies in the surfaces of those containers, rather than the air kind of blocking things. Speaking of those containers, let's talk a little bit more about the practicals of putting things in the autoclave. You can autoclave all sorts of things. So you can autoclave liquids as well as solids. So pipette tips, glassware, things like this, as well as these liquids. So bacterial media, you have LV agar. So when you're pouring those plates and things like this. Note that in all these pictures, I've only filled the containers about halfway full, or at most like 3/4 of the way full. This is because you don't want things to boil over. You want to prevent them from overflowing. And in order to really prevent them from overflowing too, you want to make sure that you're using a liquid cycle. What's going to happen in the liquid cycle is at the end of the run of the autoclave, it's going to lower the pressure. And if you were to lower the pressure really, really quickly, what's going to happen is that those molecules still have that really high energy, or at that really high temperature. You're going to be above the normal boiling point. Remember, with the high pressure, the steam is going to be above the normal boiling point. And so if you lower that pressure, you're going to just get it all to you're going to get everything is going to boil, because the vapor pressure, the outside pressure, the external pressure is going to be much lower. It's going to lower that boiling point back down, but the temperature is still above that boiling point, and so everything is just going to boil out. You don't want this to happen. So what you do is you want to, if you have liquids, you want to use the liquid cycle. And note that you can use the liquid cycle if you have solids too, but if you have you always want to make sure that you have the liquid cycle if you have any liquids in there, because it's going to lower the pressure slowly. And by lowering that pressure slowly, basically the molecules, you're going to have time for things to start cooling off, so that you get to below that normal, or you get to that normal boiling point by the time that the pressure is lower. And this way you're not going to get things boiling over or boiling out. But you're still likely going to get a bit of boiling, and so you want to make sure that your bottles and glass and things like this aren't too full, so that the stuff doesn't come out. And you also typically put these on like a little shallow tray. You don't want that tray to be too deep, or else it's going to prevent the even distribution of the steam, and it's going to prevent the drying out and things like this. But you can have a shallow tray. Sometimes what people do is they stick a little bit of liquid in the tray to help kind of make the heating more even. When you have those bottles, so you might say, okay, well, I want to make sure that I'm not going to spill things, so I better put those lids on tight, right? Wrong, wrong, wrong. You want to make sure that those lids are loose. The lids need to be loose because what's going to happen is if they're not loose, you're going to get this buildup of pressure and then they can actually explode, or they can at least get the lid stuck on there. What happens is that thanks to the ideal gas law and the rearrangement of it, called Gay-Lussac's law or Amundsen's law, what this says is basically that if you have something at a constant volume, what's going to happen is that if you increase the temperature, you're going to increase the pressure. And so basically what you can think about is those gas molecules are going to be trying to escape and get free from one another, but they can't expand. They want to get farther away from each other, but they can't. And so instead, they're just going to keep trying to move around and keep banging into one another and banging into the walls of the container. And so the pressure increases. And if you have the pressure increasing in a closed container, well, now you're going to get this really, really high pressure, and you're going to have problems where you're going to have things explode, or the pressure is going to get so high that the cap is kind of going to get permanently stuck on there. So you want to make sure that your lids are loose.
Usually what I do is I screw the lid on all the way and then I turn it like a quarter turn to the left to loosen it just a little bit. Not too much so that things don't boil out, but enough that the lid isn't going to get stuck on.
One time I was afraid of having things get stuck because of high pressure, so I made them too loose and then things spilled over. So yeah, it's a careful balance you need to make, and typically about a quarter turn is good.
You also want to make sure that you don't tighten the lids back up until the bottles are cool, because you're still going to have that higher pressure. And what's going to happen is you kind of get this suctioning effect where the lids are going to get stuck on there if you try to screw them on before it's cooled down.
Before you stick those bottles or whatever you're sticking into the autoclave, you want to stick on autoclave tape. This is a heat sensitive tape that what it's going to do is it's going to change color when it gets hot. This is really, really helpful so that you can see if things had been autoclaved. But it's not perfect. So basically it tells you if things got hot, but it doesn't tell you how long they got hot for. And it doesn't tell you that everything in there got hot for the same amount of time and for the same like thoroughness. Basically, if you think about it's kind of like a yes or no, like did it get to this hot point? But in order to actually sterilize things, you're going to have to keep things at that hot point. And so typically this is about at least 15 minutes or so, and it's going to depend on what you're trying to sterilize as well as what types of things you're trying to kill and all that good stuff. You want to make sure that you actually were at that temperature long enough, which the autoclave tape can't tell you. And so you kind of have to trust it, or there are different methods that you can use if you really, really need to make sure that things got sterilized. You also want to stick the autoclave tape as close to the center of something as possible, if possible. Basically, it's kind of like a microwave where the outside is going to get hotter first. And so if you stick something just on the outside and like the far outside of something, then basically what can happen is that's going to get hot. So the autoclave tape is going to change color, but the insides of things weren't going to get hot enough to actually sterilize things. If anybody's looking for a cool idea, I really think that they should make autoclave tape with like jokes on it. Or what I was thinking of yesterday is if you had some sort of like machine print or 3D printer, not 3D printer, but a tape printer where you can print the date on it. And that way, like when you autoclaved it, it would have the date written on it. Like, so you knew when you autoclaved it and it would like turn color. And so it would also do that. But yeah, I have no idea how anyone would do that, and not from a commercial scale or whatever. But I think that'd be cool. Anyway, when you're autoclaving things, they're going to get really hot, which is kind of a big part of the point. But you're going to want to make sure that you're being very careful when working with them. So you want to make sure that you're wearing autoclave gloves. So you have those big gloves that are going to make it hard to do things, but also make it hard for you to get burned, which is nice. Sometimes what you're doing is you're actually going to be doing something where you need to add antibiotics, such as your autoclaving the LB agar, so the stuff that you use to make bacterial growth plates, those like petri dishes that you grow bacteria on, they're filled with this LB agar. So LB is the bacteria food, and agar is this sugary gel we can use in order to make a matrix to grow the bacteria on. Normally we're doing selective growth, so we're adding an antibiotic to prevent things from growing unless they have the antibiotic resistance gene that we've programmed them to have as a selective marker to see if they also have something else that we've put inside of them, such as a gene for a protein we want expressed. The antibiotics, if you were to add them to the really hot media, the antibiotics are going to be deactivated. And so what you want to do is you want to wait until the media has cooled off a bit, at least cool enough to kind of like touch it without those big Elmo gloves. And then you can add your antibiotics and then pour them quickly before things solidify. But there's the antibiotics as well as some other things that you don't want to add before you autoclave, because the autoclave is going to inactivate them. So make sure that the things that you're using are going to be compatible with with the autoclaving. Speaking of which, you want to make sure that your containers are as well. So don't try to autoclave things like waterproof things or easily meltable things like those made of polyethylene. So you might want to look at the materials and the website and the guide and all that stuff to see what you can and can't autoclave. But you can autoclave a lot of different things. So we typically autoclave pipette tips, we autoclave Eppendorf tubes, different types of tubes, liquids, water, all sorts of stuff. Remember though that after you autoclave things, it's easy for them to get non-sterile again. So for the bottles and flasks, what we often do is you have those lids or you have foil that you can put over the top, and the inside is going to stay clean, and that inside is what you really care about. But for things like tubing or surgical equipment, you need to make sure that it all stays sterile. And so you stick them in a sterilization pouch, which is kind of like this disposable paper plastic pouch. It provides this nice little sterile bubble for things inside when you expose that pouch to the outside world. And so you'll stick things in pouches in those cases. Now let's talk a little bit more about the details of what's actually going on mechanistically inside of an autoclave. And I should mention that there are different types of autoclaves, and the exact details are going to differ depending on the type and the make and the model and all of this stuff. But this is a general overview of how some of the common ones work. So what you're seeing when you open the door to the autoclave is you're seeing the basic like oveny part, this pressure chamber. But surrounding that is going to be a steam jacket, which is going to be this water compartment, as well as various valves that are going to let the steam into the chamber, going to let air out and the old steam out. As I mentioned before, it's really important that you get that air out, because what we want is saturated steam. We just want there to be water vapor in there. We don't want there to be all that other good old stuff that's normally in the air, that dry air, that's like the nitrogen and all that stuff. We want to push that all out before we pump the steam in, or as we pump the steam in, so that we can reach this really saturated environment where we're going to have that maximal heat capacity. So when we get that condensation on the surfaces, we're going to get the biggest bang for our buck in terms of energy release. And so that we're going to be able to allow that steam to penetrate into all of those nooks and crannies of the surfaces of what we're sterilizing. In order to get rid of this air, there are a couple different strategies. One is gravity displacement. So this is a common one where basically you're just pumping in the steam. So in the beginning, you start by pumping in the steam and you have this outlet open. And what's going to happen is that this is going to make it so that the air is going to get pushed out. This steam is going to be lighter, so it's going to kind of be on top of the air, and it's going to push the air down and into that outlet valve and get rid of that air. With a vacuum displacement autoclave, you're going to use vacuum pumps to actually suck the air out before you start sending in the steam. In order to send in the steam, they typically have an electrical heater hooked up to that steam jacket. And that's going to provide that heat energy that's needed in order to get the water to boil into steam that enters the chamber. And so remember, heat is the energy that's transferred. And so you're transferring energy to that liquid And that's going to make it so that the liquid is going to evaporate. It's going to boil and get you that steam that can enter the chamber. As it starts entering the chamber, if you're in the beginning, if you're using one of those gravity things, it's going to be pushing the air out through that discharge trap. And once all the air is pushed out, that tap is going to get closed. And now when you keep pumping in the steam, the pressure is going to build. And once it gets to where you want, typically about 15 pounds per square inch or 1.1 kilograms per square centimeter, now you want to actually, you're going to start doing your sterilization or for your major sterilization. And so a safety valve is going to open up. And this way, if you add excess steam, that excess steam is going to get let out. And you reach this period where you call the holding period, where you have a steady pressure temperature. And this is a time that you want to make sure is at least 15 minutes or so. So although you only typically need about 15 minutes to get that basic sterilization, this actual cycle is going to be a lot longer because you have to have that buildup of the pressure. And then afterwards, you have to have that release. Then the heater is going to shut off so the chamber can start to cool off and the steam gets let out. And this is where you have to go slowly if you have liquids in there. Because remember that the boiling point is going to depend on the external pressure. And so if that pressure is high, you're going to have a higher temperature in order to boil. But if that pressure drops a lot, well, now it's going to be easier for things to boil. And if they're still really hot, they're going to be above the new boiling point. And therefore, they're going to boil and all your liquids would boil out. And you don't want this to happen. And so you want to make sure that you're using that liquid cycle, and just in case that you don't have your bottles filled too fully. You're going to have to let things cool off a little before you go and you try to do anything. And make sure you wear those big Elmo gloves when you take things out. Leave them to cool until you're ready to, until you're able to like touch them and things like this. Some places you might have a drying oven where you can stick things into to dry them better. Other places you just kind of stick them on your bench and wait. So you don't want things to be in like too deep a dish or anything that's going to prevent the evaporation and prevents the drying off and things like this. And if you're pouring those LB agar plates, you want to wait until they're cool enough to kind of touch, put in your antibiotics, and then you can pour them. You can also, with LB agar, kind of like microwave it again later if you're not ready to pour the plates right away. But in any case, don't add your antibiotics to the boiling liquids. And don't try to touch things that are too hot. Be careful, use those safety gloves and things like this. And hope that helps you understand how an autoclave works.